I'm going to stick with the shorter bios and introduce Dr. David Pepin, who is a professor at Harvard. He is doing a lot of work in oncology using malaria inhibiting substance as a treatment for ovarian cancer, and they identified the potential of using this same substance as a contraceptive or as a sterilant for dogs and cats, and has been doing some really interesting work in cats that he's going to present. So, Dr. Pepin, please come up.
I get a lot of funny looks at my workplace for working on cats because I'm in pediatric surgery. I work in a hospital, and never in a million years would I have thought I would be working on a contraception, and especially in cats. But I'll tell you a little bit of a story of how we got there. And I'm very excited to present for the first time my work on cats here, our work, I should say, and the first result of an in-cat trial, pilot trial. So we've been using AAV9 to deliver the malaria inhibiting substance, also known as AMH, to try to do permanent contraception in cats. And you may, I think well, AMH is used a lot, or MIS, same thing, is used a lot in the clinic in women to measure the ovarian reserve. So that's the amount of eggs you have in fertility clinics. But I think vets also use it to make sure that gonadectomy was successful. So it is used a little bit in the veterinary setting. So I feel a little bit like an imposter. I'm not a vet, and I know very little about cats, although I'm learning as I'm going. So AMH. So AMH or MIS is a hormone produced by follicles, specifically growing follicles, and by the granulosa cells. And it feeds. It's thought to feed back into as a negative inhibitor of activation of primordial follicles. So let's see if I can. Yeah. Oh, I'm sorry. I'm going to point on this side. So the granulosa cells of growing follicles produce MIS, and it inhibits the activation of primordial follicle, and also inhibits the response to FSH. So it acts on both sides. Sorry, over there. But the truth is, this was mostly theoretical, and very little was known about the role of MIS either in the ovary or the testis. There was a really good example, or a really good hint of that role from the knockout models in mice. So both knockouts of the ligand, MIS, or its receptor, MISR2, have a similar phenotype. And basically, the ovaries at birth look normal, and the only difference is that they age a little bit faster. So they run out of follicles a little bit faster, which tells you that it's a moderator of activation. So you deplete your reserve a little bit faster. So if you look at 25 days on the left, I know it's a little bit hard to see, but the two columns, there's the same amount of primordial follicle. And then when you get to 4 months, there's a little bit more depletion. And by 13 months, you run out of eggs in the knockout, whereas you don't yet in the wild type. But it's relatively subtle. And then our thought going in, although as I'll explain, it was a little bit backwards, but it was to use MIS as a contraceptive. And unlike all other hormonal contraceptive that act either on the pituitary gonadal axis, so either GnRH agonist, superagonist, or antagonist, or sex steroids, this would actually act on the first step, which is the activation of the primordial follicle. So it would be completely different from other contraceptive methods. And also, by virtue of completely eliminating folliculogenesis, you would have it would essentially look like a gonadectomy, because you wouldn't have all the hormones associated with folliculogenesis. And this is the viral vector we produced in collaboration with Guangping Gao at UMass, and we did some modifications. So originally, we were working on the human vector, and we were working on it as a treatment for ovarian cancer. And so I'll talk about the cat version a little bit later, but the initial data was using the human vector. And we did some modifications to it to make it more biologically active. So MIS is a secreted protein and needs to be cleaved to be active. And so we modified the cleavage site, and we changed the leader sequence to increase secretion. And what you see here are rat urogenital ridge And you have the Wolffian duct, which gives rise to all the male ductal structures like the epididymis, the vas. And then at the tip, you've got the Mullerian duct, which gives rise to the fallopian tube, the uterus, the cervix. And when the Mullerian inhibiting substance gets its name from inhibiting the formation of the Mullerian duct. So the best way to test it is to do this ex vivo on fetal urogenital ridge cultures. And when you put MIS, that basically that Mullerian duct regresses and it disappears. So this is a test that we use, and I'll keep coming back to it, but this validates biological activity, and it's very specific. So no other hormone, no other chemical can do this. And then we opted to use AAV9. So there's various different AAVs. Guamping Gao, our collaborator at UMass, is a proponent of AAV9. It's very good at infecting muscle, it has very high efficiency. And AAVs in general give high expression and are not very immunogenic, so they're a very good vector for gene therapy. And he had very good data showing expression, stable expression for 9 years in primate. So after you put your initial dose of virus, you essentially have a lifetime production of the protein. So this was very attractive to us as a permanent contraceptive. And some of the work I'll show that I started was finished by Motohiro Kano, who was my postdoc, who just now actually started his own lab in Japan, so that it's spreading. As I said initially, we were actually testing this virus for the treatment of ovarian cancer. And my background is actually in reproductive biology, but I was here in Boston doing my postdoc at the time on ovarian cancer. But it was still fresh in my mind, the knockout model that I showed you previously from the introduction of my thesis, where I looked at all the hormones that regulate folliculogenesis. So out of curiosity, I said, why don't we look at the ovaries of the animals that we're treating for ovarian cancer? And I opened up my first animal, and then you can see the size of the ovaries of those that are treated with AAV9 MIS. And I knew we had something big on our hands. So all the tests previous to that were done on cultured ovaries with relatively low amounts of MIS, and they had shown relatively modest inhibition of folliculogenesis, of activation, maybe 10 or 15%. But what we had here were basically ovaries that were the size of newborn ovaries. So just to give you a little bit of a background of how this works, so we give the virus here at day 0, and within 24 to 48 hours, the circulating MIS, the MIS begins to be detectable in the circulation. Essentially, it stays stable. So we've gone up, a year or more, and it basically doesn't waver. And the tissues that are infected, there's a couple of examples here, but essentially are muscles, and to a certain extent, there's a couple of other target organs like the liver that end up producing the protein as well. And if you look at the composition of the ovary, and I'll show some pictures after, there's actually the exact same number of primordial follicles. So we didn't kill the eggs. The eggs are still there. They're exactly the same amount of eggs. But there's no active folliculogenesis. So there's a few primary follicles, but no secondary or enteral follicle. So they're there, but they're not growing. We essentially completely stopped their activation. And if you look at the cross sections, this is a regular mouse ovary. These are at the same scale, so you can see how different these are. And this one was treated with AAV9 GFP, and you see growing follicles, follicles of various stages, primary, secondary, antral. The ones that were treated with AAV9 MIS have absolutely no growing follicle, only primordial follicle and a couple of atretic follicles. So as you would, we're quite excited about those results, and I remember after seeing that first pair of ovaries, I hardly slept that night just thinking of the possibilities of what we could do with this. And one of the applications was this potential of permanent contraception in pets. So one of the questions was, then what happens to the hormones if you stop the ovary? And I think you can all guess what happens. Essentially, if you look at cycling in mice, so you can do it through vaginal swabs, and based on the cytology, you know where in the cycle they are. And so a cycle basically is represented by the line going up and down, going through proestrus, estrus, metastrus, diestrus. And those that are treated with AAV9 MIS, within a month or so, stop cycling. So as you would expect, there's no active follicles, they stop cycling. But keep in mind that it takes about a month. So this is consistent with the idea that it stops activation of follicles, but doesn't really do anything to the ones that are already growing. So you need those that have already started their maturation to actually finish their maturation, and then there's no new one going into the pool. So it takes about a month to have the complete ovarian quiescence. This is what it looks like. I think these are mid-stage animals. So just after they've been contracepted for a couple of months, as you would expect, gonadotropins are up. Essentially, it's like kind of mimics castration. Steroids go down, so testosterone, estradiol are significantly lower, although they do not go to zero, and that's kind of interesting, something we can discuss later. And inhibin essentially goes to zero, so there's no growing follicles. And then we've done various mating studies. I'm showing one here, but essentially it all looks the same. It pretty much has a 100% success rate in both mice and rats. So these are 10 pairs of animals, either where the female was either treated with AAV9 MIS or with an empty vector control. And you see this is so this is the cumulative number of pups per female. So the line goes up with every additional litter. And so those that are treated with AAV9 and GFP, obviously over 6 months, produce a lot of litters. And those that have been treated with AAV9 MIS, they'll produce 1 litter, and then it's over. It's complete contraception. And again, that's consistent with the idea that you stop activation of follicles, but you actually, those that have already started growing initially at the time at which you treated, those complete their maturation, they are ovulated, and the pregnancy goes to term. And actually, those pups are completely fine. There's no sign of any effect. And the malaria-inhibiting substance actually doesn't cross the placental barrier, so that it doesn't affect the development of the pups. So that's great. We were pretty confident enough that we wanted to go in cats with this. The problem was that, unfortunately, there was a big hole in the cat genome project right where the MIS gene was. And so for this to work, you have to put the right gene in the right species. So the human gene is fine when you're working with inbred mice because they're actually not very good at mounting immune responses, but you can't go with a human gene into a cat or into a dog, so you have to make specific constructs for those animals. And unfortunately, we were missing 400 to 500 base pairs right in the middle of it. So we tried very hard to sequence this. The GC content we estimated at the time was around 93%, which basically makes it impossible to sequence through conventional means. We tried next generation sequencing using the SMART sequencing, single molecule, with Pacific Bioscience machines. They couldn't do it either. We were able to get a little bit of sequence at the 3' end, but we just couldn't get into the middle of it. So then we decided to use evolutionary genetics to try to reconstruct what the sequence should look like based on all available sequences of carnivora species. And so we basically reconstructed it and thought, hopefully we're close enough that we won't get an immune reaction. As I'll show, that as a little bit of wishful thinking. So this is what the reconstructed sequence looked like. So this is now the protein sequence. So in red was basically the sequence that we had, that we were confident in, that was in the cat genome project. In yellow is the sequence that we reconstructed through homology of carnivora. And then in green was the sequence we were able to actually sequence, the only little bit that we were actually able to manage to sequence. And even in that little bit, we actually saw some variable sites that were different between cats and other carnivora species, which we thought was a little bit surprising. But we were hoping that we would be close enough. And then, unfortunately, you can't do viral packaging if you have a high GC sequence. So we had to re-engineer the gene itself to change the codon usage to use as little GCs as possible. So we basically changed every codon in the gene. We managed to get the average GC content around 67%, which is low enough to allow viral packaging. And then we made sure that with all our modifications, this thing still worked. So we produced recombinant protein in Chinese ovary hamster cells. and we call it FCMIS, and it's compared here to the human gene, the human protein. And then the important part is that A, it's produced, and 2, that it's cleaved. So what you see at the bottom is the cleaved C-terminus. That's actually the active portion. So we knew we were able to make it, and it was cleaved. And then we tested it in the rat urogenital ridge assay that I introduced earlier. So again, a cross-section of urogenital ridge shows the Wolffian and Mullerian duct during fetal development. That's at embryonic day 14 and a half. And then if you incubated ex vivo for 72 hours with conditioned media, you can see regression of the Mullerian duct. So showing that we can not only produce this protein, but it's actually biologically active. So then we made a test batch of AAV9 with this FCMIS, this new cat construct, sort of a hybrid construct. And we tested it here intramuscular because from our feedback, this was the best route for vets. For the potential future use would be to do intramuscular injections, although all our previous work was with intraperitoneal, so we want to make sure this worked. And we've got a range of different tissues here, but again, you should focus mostly on the C-terminus because that shows you that it's cleaved and active, and there's quite a bit, mostly coming from the muscles, and especially large skeletal muscles. So we injected in the leg, and then you can see there's a ton of it being made in the leg muscle, but actually it spreads systemically, and it'll go to, you see this production in the abdominal muscle, there's a little bit in the kidney, a little bit in the spleen, a tiny little bit in the pancreas, some in the liver, and then the last one is the positive control, is the human protein. So intramuscular injection worked quite well. It's produced all over the place, but mostly, given the muscle mass, it's probably mostly coming from the muscles. And it is cleaved, which is very important. And then, so as I mentioned previously, inbred mice are more tolerant to foreign antigens, especially when you give gene therapy. It's something we can discuss also later on. It seems to help tolerance of antigens. But you can see here the effect of the cat MIS in a mouse. So this is cat in mouse. The ovary that I was given AAV9 is really teeny tiny. This is the uterus and oviduct. And this is at the same scale. This is an untreated ovary. So you can see the difference. The same result as we got earlier with the human into mouse. This is the cat into mouse. So it's also very effective at stopping folliculogenesis. So then we began our trial, a pilot trial in collaboration with Bill Swanson, in 3 female cats that were around 3 kilograms. We did an initial dose of 1.5 E13 particles per cat. We did it through intramuscular injection, 2 intramuscular injection of 1.5 mil per each shoulder muscle. And Bill did a tremendous job in monitoring the health and the cycling and the well-being of these animals. And we made sure that they produced MIS and tried to see what kind of effect we would get. And so the dose we chose as the minimal effective dose that basically the minimal effective dose that we had seen work in mice. And basically what we wanted to ensure is that we'd get more than 250 nanogram per ml of circulating MIS. And it worked tremendously well, at least in 2 of the 3 cats. So you can see production. So this is the circulating level. So the baseline is in the low nanogram per ml, maybe 20 to 40 nanogram per ml. In a normal cycling female cat, based on this assay. So different assays will give you slightly different results. And then you see production of the protein, and this is going 240 days, but I would expect this to be fairly stable for the rest of the life of that animal. And so we had 2 cats that stayed above the contraceptive threshold. What we thought was a contraceptive threshold in mice, it may not translate exactly in cats. We're learning as we're going. And one in which actually went back down to the baseline, where we suspect there's been an immune reaction to the transgene. So we looked at various different hormones, including in cat inhibin, and you see a reduction in 2 of the cats. There's cat gonadotropins seem to go up a little bit. C-reactive protein, which is as a measure of inflammation. And you can see about a week after injection, there's an increase in C-reactive protein, but then it goes right back down to baseline. So the cats didn't appear to be bothered at all from the treatment with the virus, which is a very good sign. And it is a relatively low dose when you consider what's being investigated in humans, for example. It's not unreasonable. So Bill and Lindsay did some fecal monitoring of steroid hormones, so both progesterone and estradiol, so previous to the gene therapy. And so you can see the peaks of progesterone and estradiol marking the cycles of the cats. Then you can see the gene therapy at the bottom, the timing, and then we still have a few cycles following the administration of the virus, just like we saw in mice. And then you see very significant suppression of cycling and of sex steroids for about 60 days. And then you see they resume cycling. And so we're both excited about the results and also puzzled as to why they return to cycling. And so this is the results in the 3 cats. You can see all 3 cats experience a pause in their cycling of around 60 days. The third cat, which was the one that had trouble maintaining levels of MIS, had a slightly shorter pause. But in all 3 cases, there was a significant reduction in estradiol and progesterone when you compared that suppressed period compared to previous cycling or even after the post-cycling. So fortunately, we have a lot of experience doing this in mice and rats. And we had seen some previous examples when we engineered some modifications to the transgene of immune rejection. There's 2 ways to see it. So sometimes the circulating levels will basically go down to 0, as we saw in the 3rd cat. Sometimes the levels are maintained, but then there's a loss of activity. So presumably there's a neutralizing antibody. And so this is what we suspected was happening. But then we thought we had to go back to the drawing board and try to re-engineer this thing. But again, we faced that problem of not having a genomic sequence of the cat. But then we got really lucky in that there was another draft of the genome, version 9.0, which actually covered that hole. So now we have the proper genetic sequence, and you can see that actually we introduced 30 different mutations by trying to fix this gene through homology. So it turns out that the cat is very different from every other carnivorous species. In fact, the MIS gene in the cat is different from all other mammalian in having 2 extra amino acid tracks. So basically, the C-terminus and the portion close to the C-terminus is extremely well conserved across species. So we were really surprised to see the really divergent evolution here. I don't know what it means. It's something that may be interesting to focus on for evolutionary biologists in terms of why this reproductive hormone is so different in the cats from other species. But in any case, there was a lot of differences from our transgene to the endogenous version of the gene, which we were now able to fix. So we went back to the drawing board, did the proper version of the cat MIS gene, and we just got these results a couple of weeks ago. We produced it again in Chinese hamster ovary cells. That's the sort of the cell line that everybody produces biologics with. We produce also a flag tag version so we could have efficient purification. And then we tested this new construct, and again, it's biologically active, so we can cause regression of the molar duct. So I think we've fixed the main problem, which was the 30 amino acid differences between the wild type and our engineered homologous protein. And hopefully this time, it won't be 60 days. We'll get permanent contraception if we can circumvent the immune reaction. So we've designed another trial with 9 females now. Hopefully they'll be around 3 to 3.5 kilograms each, which allows us to use a little bit less virus. So manufacturing of the virus is still difficult, but hopefully it's something that will get better as more therapies are introduced into humans. And then we're hoping to test at least 2 doses, so we can find either go higher or a little bit lower, depending on how the manufacturing goes. And then the idea would be essentially to do a very similar study where we would do shoulder injections, intramuscular, look at and monitor the health, the general health, monitor the cycling through fecal steroids, make sure that the MIS levels are maintained, look at folliculogenesis through other hormones like inhibin and gonadotropins. And then this time, we'd really like to go to mating trials, assuming that we see evidence of loss of cyclicity. So there's a lot of people that I need to thank that were involved in this study. Nick Nagy-Carey, who is a technician in the lab that helps me produce the ELISA and the cat protein. It was very instrumental in making these different proteins, which is a lot of work. Kano, who's at the back, who did a lot of the original work in mice and also helped the work with the new cat construct. And our collaborators, Patricia Donahoe, who was instrumental also in teaching us about the malaria inhibiting substance, having cloned the hormone itself in the 70s. And Guanping Gao, who is a master of gene therapy at UMass, and Bill Swanson, who obviously we couldn't do this in cats without him, and the founding from the Michelson Found Animal Foundation. And although I didn't show any of the work, but we're also funded by the Bill and Melinda Gates Foundation to do contraception in humans as well, with the same idea of targeting the initial step of activation of the primordial follicle. So thank you.